I realize I'm beginning to be a bit boring, but I would like to bring home a couple of major conclusions, one of which is that I just tried to illustrate. It pays to look at the diversity in nature to learn about general phenomena. So, species diversity. Now I will discuss in the first half of the second hour today an observation that would really puzzle you if you are not following the field very closely and if you are studying immunology, and you probably wouldn't even believe me if I would give this without any introduction. Another fish species turned up a huge surprise in terms of how the immune system operates, and that is not now about some special or specialized function of one or the other gene. It's about really a system or structure of the system. So in order to explain this, I have to remind you what you probably all know already. So bear with me to go through this introduction just in case. How T cells recognize antigen. You probably heard and you know that antibodies, and we discussed that in the beginning, antibodies can recognize almost any structure with great specificity and great discriminatory capability. T cells have a similar level of discrimination. However, this is not focused on any chemical structure, certainly to the first approximation, but on peptides, that is, on breakdown products of proteins. And these breakdown products of proteins are being displayed to the T cell receptor through a protein or class of proteins that is called MHC. And MHC stands for major histocompatibility complex encoded proteins. It's a complex genetic complex, and we come to this a bit later. They come in two different forms, MHC class 1 and MHC class 2, and you probably all know this. And in the meantime, people have worked at looking at the structure of how a complex of the antigenic peptide sitting on top of the MHC presentation molecule and the T cell receptor look like in 3 dimensions. And this is now a cartoon that shows you this. In fact, it's an incredibly complicated structure. So here we have a T cell receptor. This is this part here. Oh, sorry, this part. The T cell receptor is green. So it has, if you remember, its origin in the assembly in the soma of VDJ elements. They are being put together, and this protein then faces the antigen. The antigen is buried in a binding groove of the MHC molecule. The MHC molecule also is a transmembrane protein, so it's anchored in the membrane of the antigen-presenting cell. The MHC binds the peptide, but it binds it such, and we'll come to this in great detail a bit later, that some of the amino acid side chains of this peptide are exposed to the outside world for the T cell receptor to see it. And the T cell receptor does not recognize the backbone of the peptide chain, but rather the side chains, because they make the identity of the amino acids, as you know. So they point towards the T cell receptor. The T cell receptor in its variable part, particularly the complementary determining region number 3 that we mentioned yesterday or the day before. So it makes contact, but it also makes contact to the MHC. And the T cell receptor also needs signaling components to tell the T cell the receptor has seen the antigen, it makes a conformational change, and that is transmitted into the cell, so the cell knows, yes, yes, we've seen our antigen. But what is more, the T cell receptor, or the T cell rather, needs a so-called co-receptor that also comes in 2 forms. Here in this particular example, it's the so-called CD8 form. There's also another form, it's called CD4. It's a molecule that is sitting in the membrane here, and it extends across this entire complex and binds to MHC. It's basically like a clamp. And the important biology behind this is that the co-receptor binds a protein kinase at the inside of the T cell that is capable of activating pyrosines in the signaling parts of these signaling molecules. So we're not discussing this intracellular signaling in detail. You can read about this in the textbooks. Suffice it to say for our discussion that the T-cell receptor is binding the antigen that is complex with the MHC, and the co-receptor helps decide which MHC type is presenting to the T-cell receptor. And the selection is a complicated process during T-cell development. We're not discussing that in detail, but you will get the idea of the whole system in a moment. Because the antigenic peptides can be derived from two different sources. As you know, when proteins are synthesized in the cell, most of them don't fold properly, and they are immediately degraded and recycled. About 30 to 50% of freshly synthesized proteins don't fold properly, are immediately degraded, and the degradation occurs through the so-called proteasome. Proteasome is basically a protein degradation machine that takes proteins and chops it up into small bits, and these bits are called peptides. So the protein goes in, is being shredded, and these peptides are being liberated. Now, for the cell, these peptides are quite a useful resource, because the peptides are further degraded into amino acids, and these amino acids can then be used for biosynthesis once they've gone through all these required steps. So it's basically a recycling machinery. This is the waste bin. The shredders it. Peptides are being degraded into amino acids, and then it goes into protein synthesis. Some of these peptides escape complete degradation, and they are being transported by a specialized transporter associated with peptide presentation. This is why this complicated names comes from. It binds these peptides and it channels them into the ER, where they are then not degraded because they are binding. It's a complicated process, but the principle matters. They are binding to the empty grooves of these MHC proteins. Once they are sitting in this groove, they are basically protected from endo or exopeptidases, and they can remain there. So then they become stable. So basically, taking peptides from the cytosol through the transporter into the ER and loading them on these empty binding pockets of these MHC molecules is a way of protecting them from degradation. Now, this must be important, otherwise you would not sacrifice energy and resources that you would normally be able to recycle and use again for protein synthesis. And the reason why this is selected for and maintained is because these complexes, peptide and MHC, travel through the Golgi to the cell surface, and at the cell surface, basically, by presenting these peptides, tell the outside world about the ongoing type of peptide synthesis or protein synthesis in the cell. That is, of course, interesting and perhaps not so terribly useful when the cell is normal, but it becomes very important when the cell, for example, is infected by a virus that replicates and makes proteins in the cytosol. So viral protein often also does not fold properly. Viral protein is degraded, peptides are escaping complete degradation, are being loaded, and all of a sudden up there is not a self-peptide, but a viral peptide. And because, and we come to this a bit later, because the T-cell receptors, or rather the T-cells expressing them, have been educated to tolerize or to be tolerant to self-antigen, they become activated when they all of a sudden see a foreign peptide. So this is basically monitoring what is happening inside the cell, and it can be monitored from the outside. And this will become important when we look at alternative functions of the MHC system. And this is why I was referring to that sex is important, because this is not only for immunology, but that's also for sexual selection. So these peptide MHC complexes are seen by the T-cell receptor. The T-cell receptor can read these side chains and determine the sequence of the protein. And because it knows, as it were, through selection which sequences are self and which sequences are not self, it will react to those that the repertoire has not seen before. And this is the so-called class 1 pathway. MHC class 1 molecules bind these cytosolic peptides to a first approximation and present it to the cell surface in this particular structure. There's a second pathway which degrades proteins. This is the so-called lysosomal pathway that you all know about. So it is about taking up proteins, either cell surface proteins or exogenous proteins, encapsulating them in vesicles, so-called endosomes. These endosomes then fuse with other specialized organelles in the cell to create an environment that is conducive to protein degradation, and the same thing happens. These proteins are degraded by lysosomal proteases, and these peptides can then be bound to a second type of MHC molecule by the name of class 2. You see that process here, and this goes up to the cell surface. And now this peptide that's in that binding groove is indicative of what the cell has taken up from the outside or has recycled or is in the process of recycling from the cell surface. A second pathway of proteins, not only cytosolic but also lysosomal proteins, appear at the cell surface. Now, the T-cell receptor that sees MHC class 2 bound peptides requires a different type of co-receptor, not the CD8 that we've seen before, which is associated with class 1. It requires CD4, a structurally quite dissimilar protein from CD8, but it has a similar function. It is expressed by the T cell, and it bridges this complex and binds, goes from here to here, binding to the MHC class 2. So that means that MHC class 2, which mediates peptide recognition through the lysosomal pathway, although there is some crosstalk, which I'm ignoring at the moment, and a special co-receptor that targets or guides the T-cell receptor to these MHC class 2 peptide complexes by the name of CD4, are one of the two major pathways by which T cells recognize antigenic peptides. This is the explanation how this works. I said to you that the co-receptor binds to tyrosine kinases, and the one that is important here is the so-called LCK protein tyrosine kinase, and brings it to the T-cell receptor, but because the T-cell receptor, when this engagement happens, so when the MHC peptide complex comes near the T-cell receptor, then the co-receptor can bind MHC, brings this kinase, the kinase starts phosphorylating these receptor components, and a signal occurs. CD8 binds to class 1, CD4 binds to class 2, and in that way, the LCK is always brought to the T-cell receptor and it signals. When there is no MHC, this cannot quite work, and we are not discussing this particular problem now. So these co-receptors are instrumental in telling the T-cell, your T-cell receptor has seen a ligand, because only then becomes this complex complex in close proximity so that the co-receptor can bind.
So each T cell has both CD8 and CD4?
No, in the end, in a mature T cell, it has either one. Either CD4.
MHC presented.
Yes, this is a complicated selection process which we are not discussing. We simply take it for the fact for the moment that one type of T cell has CD4, the other one has CD8, because it's been selected in the thymus for binding to either or the other MHC class, MHC type. I'll only come to this in a moment. Now, you might say this is somewhat irrelevant if a T cell recognizes this one or the other class of MHC molecule, or it recognizes or uses one or the other co-receptor, but in fact it's not, because the T cells that express these combinations, either CD4 or CD8, are functionally quite distinct. They all have the same type of T cell receptor, the T cell receptor alpha beta type, yet they are functionally quite different. The ones that express CD8 and recognize peptides in the context of MHC1 are so-called cytotoxic cells. These cells kill the presenting cell, whatever it is, and that is of course a cytopathic, if you wish, effect, and that's very important, for example, to kill away virus-infected cells. Viral proteins, for the most part, are made in the cytosol, so it makes sense that MHC class 1 restricted T cells then kill virus-infected cells. And there are a number of transcription factors that stabilize the developmental fate of these, this particular T cell lineage, a number of effector molecules that are being produced by these cells, and once they are activated, then they release certain factors, and with the end result is that these cells are killed. They need certain factors for the production of this particular cell lineage, for example, interleukin 7 or interleukin 15, both of which are required so that these cells during development can emerge. So this is a genetically programmed process, very complex. We're not going into this. It's not only genetic, but also epigenetic control that plays a role there. So it's a difficult subject that's intensely studied. For our purposes, it is important to remember MHC class 1 restricted, as they are called, CD8 plus, CD8 expressing T cells are cytotoxic. What about the CD4 ones? The CD4 ones are a really special kind. They are called helper T cells. And they are called helper T cells for the most part because they help to initiate a response of the immune system through the B cell compartment. They have some other functions, but we just take it for the moment that they help somehow shape the antibody response in the immune system. Without them, this will not work properly. And we are used to think of our immune system of using CD8 and CD4 expressing T cells, CD4 expressing T cells expressing MHC class, or are restricted to MHC class 1, class 2 presenting peptides, and they have this CD4 co-receptor. Again, there are different lineages that function in various aspects of the immune system, the most famous of which are perhaps the T regulatory cells that regulate the fine balance between self and non-self activity, cells in the follicular helper cell compartment that regulate hypermutation and the formation of the germinal center and the maturation of antibodies to be highly specific, high affinity antibodies. Various other types of factors that we don't have to discuss in detail, resulting in a rather complex diversification of this primordial helper cell type that has various subfunctions. And because all of these are very important, as attested by the clinical phenotype to which I come in 1 second, we are used to think that the immune system needs these two arms of the T cell system. What are the phenotypes? If you have genetic mutations, for example, in the effector molecules here, you cannot express a cytotoxic phenotype, which can be quite difficult. This can result in immunodeficiency, as you can imagine, if you cannot kill virus-infected cell. There are animal models, of course, but also human patients known that have defects in this particular pathway. Very bad. There are diseases where viruses specifically target and kill CD4 positive T cells. You know one disease that we mentioned before, HIV, for example. So this arm is severely impaired, and this is one of the problems why these patients do rather poorly. There are mutations in transcription factors regulating these sublineages, as it were. For example, this one here, where this transcription factor FOXP3 is mutated, and that results in a severe autoimmune disease because these regulatory T cells suppress self-reactivity. And this is a syndrome that was discovered or described in humans and then soon replicated in animal models, particularly in mice, so-called IPEX syndrome. So it's a syndrome of severe inflammation that mostly affects the intestine and then the entire body. So all of this is very important, as attested, as I said, by either experimentally induced phenotypes or by naturally occurring phenotypes. Now enter a rather famous species, the Atlantic cod. And I'm sure you've been, well, some of you might have been to England. When you go to the fish and chips shop, they sell you cod, fried cod, and chips. That is French fries, basically. Now, when the genome of the Atlantic cod was analyzed, people were really surprised, because what they found was that the Atlantic cod does not encode CD4, nor does it encode MHC class II. So you would be telling me this is not possible, because if I take out this entire thing here, we wouldn't live. And that is true. We would not live, because we are used to having this. The Atlantic cod has done away with this pathway. And it lives a happy life. The average lifespan of the Atlantic cod is about 25 years. It lives in the deep sea of the Atlantic. It's very cold, it's dark, it's full of pathogens, but it's still happy, and happy enough to be caught by fishermen and be put on the plate, fish and chips.
What about FOXP3, IL4, all the vector cytokines? Are they helper cytokines and transcription factors?
I don't know exactly, but I can tell you that FOXP3 is present. I believe all the other ones are also present. It's basically the MHC class II and the CD4 molecule that are missing.
So maybe CD8 T cells took some of the functions?
I'm sorry?
Maybe CD8 T cells just took some of the functions?
Well, here's the question. Now, how does the fish survive this? Of course, it's not a genetic accident. Okay, so there must be some adaptation, and the question now is, what is the adaptation? The adaptation is, and you can see that here, now I don't reach up there, but if you look at panel A, you see that there is the MHC1 diversity in terms of number of MHC class I genes in human and stickleback. Stickleback is another fish. They could have put in zebrafish, for example. And you see that they have, in this particular assay, about 10 genes, 10 different genes. They come in different alleles, of course, but they have 10 genes. Now, the Atlantic cod, as one way of adapting to this brutal genetic insult of taking away this entire pathway, has ramped up the MHC gene number to about 100. And now you can think about how this might be possible, that they perhaps can survive the lack of this lysosomal degradation pathway and presentation through MHC class II, perhaps by ramping up MHC class I. Perhaps there is some indication that this cross-presentation that I did not really specify, but that occurs by exchanging, let's put it this way, exchanging peptides between this classical cytosolic and lysosomal degradation pathway. So it's not quite so clear and clearly distinguished as I portrayed it. So maybe this is one way of overcoming that deficiency. And there is another adaptation. What they found was, when they were looking at toll-like receptors, now receptors that we know are germline encoded and recognize specific general classes of components of pathogens, for example, lipopolysaccharide we talked about, or some other general components of parasites, flagellin or whatever, that they all of a sudden have increased their number of toll-like receptors. You see, this is the Atlantic cod here. So certain gene families of TLRs, the TLR8 family, has 1, 2, 3, 4, 5 members. TLR7, all of a sudden, 5 members, and you can see that everywhere here. So the capability of recognizing potentially dangerous molecules in the sense that they recognize substances that are normally carried by bacteria or by parasites, So they've dramatically increased their detection capabilities and perhaps also their specificities. That is not quite known. To overcome, presumably, to overcome the deficiencies, they have to suffer from their loss of MHC class 2 and CD4. And what was known before that experiment was done, this actually was one of the reasons why it was done, although perhaps the primary reason was economy, because cod, fish and chips and so forth is very important economically for the fisheries. So they wanted to understand why this fish is capable of surviving for that long and how one could make it fit to survive in aquaculture, which is not so easy. But anyway, so what was known from immunological studies is that these cold water fish have a very slow antibody response, whereas we, warm-blooded animals, have a very vigorous antibody response. So when we are encountering antigen, our T cells help the B cell compartment to really get going very quickly, and we make antibodies quickly, and we refine the antibodies, and this is not possible here. So the antibody response is rather sluggish in these fish. It takes a long time. There's no affinity maturation. There's not much happening there. So that means that the antibody response is not terribly useful to these fish. They have to find other ways of dealing with problems. And I just outlined two that were discussed in the paper that I refer to down here. So it is possible to build an immune system that equips a particular species with a rather functional and fit immune system that lacks essential components that would, in the absence, kill us. And I think the reason why that is interesting is not only to think about the organization and design principle of the immune system by taking a look at these examples, but also perhaps to think about ways of treating immunodeficiency syndromes. For example, in humans there is a syndrome that's called Behr lymphocyte syndrome that affects a transcriptional regulator that regulates the expression of MHC class 2. And these patients do very poorly because they can't make MHC class 2 with all the consequences that I was just outlining to you. Now, of course, you can think of perhaps gene therapy to bring back the faulty gene or restore faulty gene function, but you might, of course, that is now fantasy, but you might think of using examples like this to somehow overcome the deficiencies that are caused by this particular genetic defect. Perhaps one finds ways of mimicking these adaptations in a patient to help him survive this genetic defect. So that it's just a way of intellectually interesting circumstances that one can use to think about syndromes that are probably more closer to our heart. Now, where does the MHC come from? I've simply taken this for granted now that we have MHC as a means of presenting peptides to the T cell receptor. It comes out of the dark ages, out of the blue, you might say. It has been shown that the MHC, or the genetic components that make up the MHC, this locus, emerge in a certain chromosomal environment. So one can trace the neighboring genes of the MHC. Now this is perhaps a bit difficult to read. One can trace the neighboring genes of the MHC locus through evolution. And what I did not mention, but you know, of course, that during the course of chordate evolution, the genome has been duplicated once, twice, and perhaps even 3 times. So one can look at what is called paralogous regions in the chromosomes. So from one chromosome you make 2, you make 4 chromosomes. So you can then trace the origin of an ancestral chromosome. So the MHC that has been studied, this is now human. MHC is located on chromosome 6, and you can't read this now, but this is MHC class 1 and class 2, and you can see these blue boxes here, they are neighboring genes. And now if it's true that the MHC is an invention, you can look at the paralogous chromosomes and ask the question, do all 4 paralogous chromosomes have an MHC? If they do, then presumably MHC has originated in the original single chromosome. If it's not, then it might have occurred at a later stage. Or else you make the argument, okay, it was present initially, but it was lost at the other 3 locations, which is, of course, always a possibility. So what one can see without going into the detail, these 4 paralogous regions can be identified. This is work by Masanoro Kazahara in Japan. He spent quite a bit of effort on this to delineate what he calls the proto-MHC chromosome. So he can identify the neighboring genes. He can really delineate the original region where the MHC comes from. But when he reconstructs the original chromosome, there is no sign of MHC. And the argument there is that MHC, or the genes associated with it, arose by gene duplication of a primordial gene that we no longer recognize as the ancestor of MHC. MHC also belongs to the gene family of immunoglobulin super, of the superfamily fold. So it's part of a bigger family, and there must be some proto-MHC gene that we no longer recognize that perhaps was at this particular location and then diversified through internal duplications across this complex chromosomal duplication events. Now I mentioned, and this is my last couple of minutes for today, I mentioned that the antibody response, and I come back to the emergence of new genes in the immune system then at the end today, that the antibody response in the fish is very sluggish. And we have a very efficient antibody response. And I explained to you that it's important for the activation of lymphocytes that are being presented with antigens through MHC, for example. But there is also a technical problem that concerns the volume. We refer to the blue whale as a rather big animal with a diverse repertoire of T-cell receptors, with many T-cells, but not necessarily a T-cell with the required specificity next to the site of infection. So there is this big problem of organizing and orchestrating the physical apposition of a T-cell with a desired specificity and the antigen presenting cell that presents the cognate antigen when there is infection going on. So how do you organize this as an engineer? You might simply operate by stochastic means. You say, okay, I just generate so many T cells and I ramp up the clone number so that each cell with a specific receptor is represented maybe 1,000,000 times, that the entire body is basically consisting of a mixture of these T cells, and there is never a T cell with the desired or required specificity far away from the site of infection. But that is relatively inefficient because there's not enough room to represent the entire spectrum of specificities with these many numbers of T cells. So that is not a very clever system to do. So what the tetrapods during vertebrate evolution have invented is a specialized lymphoid organ to deal with this particular problem. And I mentioned this in the very beginning. One solution to this problem is to set up traffic rules, tell the T cells, yes, you might as well, if you want, patrol the body, but why don't you just sit in a specialized compartment and wait until you are being called into action? And we defer the policing activity, as it were, to a cell type that is non-specific in terms of antigen recognition. And one particular cell type that is non-specific in this regard is, for example, the dendritic cells. They come in various flavors, but the long and short of it is that they basically sit everywhere in the body. They can take up foreign antigen, or even self-antigen, of course, but they can process antigen irrespective of specificity, but they are being told to take this up and then migrate to a certain meeting point. Okay, when you arrive at the airport where Anja came to pick me up, there's a meeting point. It wasn't quite like this, but I can simply lie to you. So there is a meeting point where everybody comes. I have to go there. You don't have to run around in the entire airport. The meeting point will bring people together. And these meeting points in the immune system are called, as you know, lymph nodes. So to organize that system is you tell the antigen-presenting cell, you do the policing, you go through the entire bodily space, you pick up antigen, you bring it to the lymph node, and in the lymph nodes we have our T cells sitting around there to a first approximation, and they know they have to go there to be activated. That is a very efficient way of dealing with this problem. So this lymph node system was invented not very early in vertebrate evolution, amazingly enough, but relatively late. It was invented somewhere here. Some reptiles have what one could call primordial lymph nodes. Some birds have what one could call primordial lymph nodes, but definitively all mammals have lymph nodes.
Lymphatics, the vessels?
I'm sorry?
The vessels, lymphatic vessels.
Lymphatic vessels are already present in fish. But the aggregated structures that we know as lymph nodes are beginning to appear somewhere here. But they're in full existence in mammals. So some birds have it, some birds don't. Some amphibians have it, some amphibians don't. But more birds, and they are more elaborate in their structure than what we know in mammals. So this is, when you look at this arrow, this top arrow here indicates general hematopoietic tissues, or bone marrow, as we call it somewhere here, or gut-associated lymphoid tissue, that is cells that are sitting in the gut wall. The thymus we discussed, the spleen as a way of filtering and organizing immune responses in the interior, as it were, and then the lymph nodes. The lymph nodes are the most recent invention. And whenever there's something that is recent and comes seemingly out of the blue, a bit like MHC, then it becomes really interesting, not only for, in our case, immunologists, but also for geneticists. Where do genes come from? This is a general problem that we still haven't solved. And I will just give you an example of how lymph nodes came to in existence, because all of a sudden genes appeared that we didn't know before that are important for the generation and the workings of these lymph nodes. So just to give you the overall picture as a reminder, shortly before the end of the day. So we are developing T and B cells. We are developing myeloid cells that will function as antigen-presenting cells. And in the end, in the secondary lymphoid tissue, we bring them all together. This is their meeting point. They interact through interleukins, chemokines, and cytokines. And these antigen-presenting cells here, just M for macrophages or whatever, T cells and B cells, happily sit together, influence each other, and orchestrate the immune response. And this is, as you all know, of course, when you have an infection and your lymph nodes swell, it's very painful. It's painful because there's an influx of cells. Cells start proliferating, and because the lymph node is encapsulated with a fibrous capsule that cannot easily expand, it is mechanically stressed, and this is what is painful. And when the infection resolves and the immune follicles then regress, then of course it's no longer painful and the lymph node becomes smaller again. So this is what the secondary lymphoid tissue is all about. Just in parentheses, you might of course ask, then how is it then possible that fish make an immune response? I mean, they also must somehow organize the interaction between antigen-presenting cells and lymphocytes. Yes, of course they do, and one can show this experimentally, but it seems a bit more dispersed. Why that is so and how that works is not yet clear, but it's of course of great interest to understand. So lymph node development, just to give you a graphical illustration of what we see. When you look at the lymph node through immunohistochemistry marking different cell types, I'm not going into any detail, I'm just illustrating this here. We have different compartments, so cell types sort themselves out. T cells, B cells, and particular stromal cells, they form particular zones and interaction sites so that the immune response can progress in a rather ordered fashion. That is very important for the immune system to regulate the immune response. So it's an anatomically well-structured organ. It requires these communication signals that also become more and more diverse as we go through evolution. And when you look at the development of lymph nodes, you even see some developmental progression. For example, you begin with mesenteric lymph nodes. This is mouse development very early on. You progress over brachial lymph nodes, inguinal lymph nodes, popliteal lymph nodes, pious patches, and then the last type of tissue that is developing after birth is the nasal associated lymphoid tissue. So there is a progression in development, perhaps reflecting evolutionary stages of secondary lymph node development, but we don't know. And what one can show through genetic analysis is that these lymph nodes are different by the phenotype of genetic inactivations. So I've listed here a couple of genetic components of this lymph node development. I'm not going into the detail. Lymphotoxin alpha here, lymphotoxin beta receptor, a kinase. This is a transcription factor, REL-A, a TNF receptor. You've heard all these buzz names. And when you then look at what is the outcome, formation of lymph nodes, all lymph nodes, pious patches, which is a tissue that's associated with the intestine, the nasal associated lymphoid tissue, then you sometimes see a minus or a plus. And then you easily recognize by going through this list of genetic changes, sometimes you can do a lot to the genome and you still generate this particular type of secondary lymphoid tissue, whereas all the others are missing. Or you can do something here, you have this plus but nothing here, or reduced, or plus here and nothing here. That already indicates to you that the genetic requirements for these tissues are different, so they must be functionally different. Now what I'm pointing out in passing is that specific genes are very important. And one important gene is, I think I have to go a bit faster now, one important gene that is important is the so-called lymphotoxin beta receptor. Lymphotoxin beta receptor is a receptor for a molecule that is called lymphotoxin, as the name would suggest, and they belong to a particular type of family, the TNF family, that is again a multi-gene family of a particular structure. But this gene, the lymphotoxin beta receptor, that regulates the differentiation of important cell types that initiate the formation of lymphoid tissues, does not exist in fish. It only appears in animals that have properly formed lymph nodes. So what does the lymphotoxin receptor do, amongst many other things? There are mesenchymal progenitor cells that can go through a differentiation pathway programmed by this lymphotoxin beta receptor, which is engaged by this lymphotoxin ligand, which also doesn't exist in lower vertebrates in fish, only arises later. So a ligand receptor pair arises out of seemingly nowhere, that programs these cells to respond to another cell type that is perhaps a little older, the so-called lymphoid tissue inducer cell type. And once these two cells interact, secondary lymphoid tissue formation is initiated. And it now really matters which mesenchymal progenitor becomes responsive to this signal, because this determines where the lymph node occurs. When you are a doctor and you examine a patient, you know exactly where to go for lymph nodes, okay? The cervical lymph nodes. You can do that yourself. So when you have a sore throat, this will become very painful. So you know where to go here. You know the bronchial lymph nodes, the inguinal lymph nodes. So these lymph nodes develop always at the same place. So there is a pre-programmed sensitivity of certain mesenchymal progenitors to respond to these signals, a rather enigmatic process. We really don't know yet what makes them susceptible to these signals to then jumpstart the formation of lymph nodes. But what is important, that this lymphotoxin beta receptor and its ligand in terms of genes do not exist in the genome of lower vertebrates. Now I'm showing you a similar cartoon to what I showed you about the MHC. Now looking at paralogous chromosomes, or rather regions, and what you can see is in frog, remember amphibians begin to develop these primordial lymphoid, secondary lymphoid structures. In this chromosomal region, there's lymphotoxin beta. So we're just focusing on this. And we now take note of the neighboring genes. The neighboring genes we can find again in the genome of fish, but no sign of this lymphotoxin beta. We go to opossum, wallaby, mouse, and human. Lymphotoxin duplicated. Lymphotoxin duplicated. So this is the same here. So this genomic region comes out of nowhere, and it doesn't exist in earlier species. And when this happens many times over, it's very unlikely that these genes have been lost, rather they emerged anew. And one interesting finding that one observes is that new genes often arise at so-called synteny breaks. That is, when chromosomes are broken apart and fused together in a new way. So the gene order, as you know, is not always the same between mouse and humans, or the great apes and humans. There is always some shuffling around. And wherever chromosomes break, very often new genes arise. And one can see that here, that synteny breaks often occur. So here's a synteny break, often occurs where these new genes arise. So there is birth of new genes. And that, of course, not only applies to the immune system, but birth of new genes is often associated with the development of morphological or functional novelties. So let's see what I have prepared for the summary. Nothing. So I summarize in words. What I was trying to tell you today is that when one studies seemingly exotic animal models for their immune functions, one can turn up quite a few surprises. Either one can make use of their particular physiology. I gave you the example of zebrafish that can be transplanted very easily. Or one finds alternative solutions to seemingly important immunological pathways, like in the Atlantic cod, that has done away with one particular type of cells and has done away with a certain type of immune reaction that we think is important in warm-blooded animals, for example. So this is my mantra. Don't only study humans and mice, look further. And I've given you this example, and we will expand on this tomorrow and the day after. So thanks very much for your attention.
When did pair stages appear in evolution?
I'm sorry?
What about pair stages? When did they show up in evolution?
Pious patches probably also occur only in mammals.
And tertiary lymphoid tissue?
And?
Tertiary lymphoid tissue.
There is a question about tertiary lymphoid tissue. This is lymph node-like tissue that occurs at sites of chronic inflammation. I would think, although I don't know specifically that this has been studied, I would think that also co-occurs with the appearance of secondary lymph nodes, because all these signaling components need to be there for tertiary tissue to arise.
And you think LTIs did not exist before, or ILC3 for the Boehm?
The question is whether these lymphoid tissue inducer cells did not exist before the appearance of morphologically discernible lymph nodes or secondary lymphoid tissues. The answer is I don't know. Yeah.
You just mentioned a little bit, but maybe you can briefly discuss a point about the role of secretory immunoglobulins like IgA in humans and IgT in fishes, their mechanisms, how they contribute to maintain the composition and structure of microbiota.
Yeah, I could now speak for another hour, but I'd rather not say much because the mechanism is not known how that works. it's just correlation and association. I think we lack key experiments by taking away, in many different species, taking away this diversity or in one way or another manipulating it and then see what happens. There are some indications that if you, for example, remove IgA, then there is no change in microbiota or no clinically relevant change in microbiota because IgM can take over. But if you eliminate somatic hypermutation, then there is a big difference in the composition of the microbiota. So there is a correlation, but we do not know what is chicken and what is egg at the moment.
Speaking of this lymph node-less organism, is there any future at all in the lymphatic vessels? Because I think the belief is that subcapsular macrophages of lymph nodes are maybe as important, if not more important, than T cells and B cells in terms of preventing spread of the pathogens.
Yeah. So the question basically targets what are the primordia of lymph nodes. So that
Or if there are alternative structures that filter the lymph and capture the...
When one looks at animals that have lymph nodes but no recognizable lymph nodes as such, sometimes there are intralymphatic aggregations of lymphocytes and perhaps also other cell types. So one believes that perhaps this was the beginning of a more organized structure. But that, of course, has not been studied genetically, so it's difficult to know.